nucleic acid amplification, which is the PCR reaction that we normally use. And the PCR reaction is a primer-directed in vitro enzymatic reaction. So primer-directed, as we see in the past few days, I always mention that we have to design the primer directed, okay? And the primer basically can also define the region of the sequence that can be amplified. And the in vitro enzymatic reaction, that's because the PCR reaction is a reaction for DNA synthesis. And so you need the enzyme, which is DNA polymerase, okay? DNA polymerase perform the enzymatic reaction to synthesize the DNA strand. And the PCR reaction, so here that we say the forward primer, reverse primer, and then this primer basically define the region, all right? Define this region to be amplified. And also the primer distance can also determine the length of the PCR product, okay? Because the distance from the forward primer to reverse primer that give you the length of the PCR product. And for a standard PCR primer, like when you try to perform some PCR reaction to amplify a specific region without any particular variation, the primer normally is only 18 to 30 base pair long, and that this DNA must be complementary to the sequence flanking to the region that to be amplified. And in our case, we generate the primer like 60 nucleotide long. That's because we introduce the mutation. We want to introduce the variation that so we have 20 nucleotide match the template, but the other 40 nucleotide tail, that's the specific DNA sequence we introduce through the PCR reaction, so that we can have this hybrid DNA molecule, and we can use that hybrid DNA molecule to knock out a target gene. So that's why we design a 60 nucleotide long primer. And also different kind of primer, like the primer that people used in the southern blot hybridization. And that primer can be even 200, 300 base pair long, because you want to identify a specific DNA region at the location of specific DNA region, so that you use a very long DNA fragment as the primer. But that's not PCR, that's the southern hybridization experiment. But for PCR, normally the primer is only 18 to 30 nucleotide long. And the distance between the primer binding site will determine the size of PCR product, okay? Because the distance between these two primer that will determine the size of the PCR product, okay? And the standard PCR reaction mix, normally you need to have a primer, like the primer forward, reverse primer, and then you need to provide a deoxy nucleotide, right? Like a deoxy ATP, CTP, GTP, TTP, because it's a DNA synthesis reaction. So you need to provide these deoxy nucleotide. And also you need a polymerase and template, all right? So these are the component required for the PCR reaction, and the polymerase that we will discuss later, okay, after we introduce you the feature of the PCR reaction. So the amplification reaction basically will be a repeat cycle of the steps of PCR reaction. You repeat it 20 to 50 cycles so that you can amplify your target. And the PCR cycle basically is a 3 steps reaction. Okay, so you have denaturation, annealing, and the extension. And the denaturation is basically, that's the first step that you need to denature your template DNA, so that you need to increase the temperature to a 90 to 96 degree. And because this temperature is so high, all right, so that when you perform the PCR reaction, this polymerase, you need to use a specific polymerase, because at the 90 degree, all the protein are denatured. When they are denatured, then they are not functional, okay? So that you need to use a thermostable polymerase, all right? So that's why we use a Taq DNA polymerase in the PCR reaction. And the annealing. So at the annealing stage, basically, it's a 40 to 68 degree, depend on the melting temperature. All right. So that's why it is important that you identify the primer sequence. Then you have to determine the melting temperature. And the melting temperature, then once you determine the melting temperature, then you do the Tm minus 2. That'll be the annealing temperature. Okay. You set up 2 degrees below the melting temperature, then that will be the annealing temperature, so that you can ensure the more than 50% of your primer will bind to the target. Once you finish annealing stage, then that will be extension. So extension, basically the 70 to 74 degree, that's the optimum temperature for the Taq DNA polymerase, so that Taq DNA polymerase can perform the DNA synthesis at this stage. So basically, just repeat denaturation, annealing, extension, these 3 steps 20 times, 30 times, 40 times, so that you can amplify your target sequence. Okay. So this is the more detailed explanation. Okay, so first step, the cycle 1, step 1. So you have denaturation. So these 2 blue template DNA be denatured, and also the primer sequence will be denatured as well. Okay, and then the cycle 1, step 2, annealing. So at this time, the temperature dropped to 2 degrees below the melting temperature. Then the primer, right, they will find their target sequence, and then they will bind. All right, when you decrease the temperature below the melting temperature, the primer will bind to the target. And then the step 3, then you can see this gray one. So from this primer, they extend all the way to the end, but beyond the border of the other primer. Okay, and then for this primer, they also extend the synthesized DNA all the way to the other end. All right, so this is the first cycle. Remember, the blue one is the original parental strand, template strand. The gray one here, that's a newly synthesized DNA strand. And now, when we enter the cycle 2, so remember, these 2 are newly synthesized from the step 1. Okay, like here, from the cycle 1. All right. So from the beginning, you have 1 template, and now end of the cycle 1, now you have 2 sets of double helix DNA. But these 2 sets, they only have half of the DNA synthesized, like this here. All right, so that'll be here. And now for the cycle 2, then you have to denature these 2, so that you have a green one, and this gray one is button here, and gray one is here. And these 2, the green one here, they will perform the exact same reaction like the cycle 1. All right, so these 2, this green one will perform the same reaction like the cycle 1. And now, if you look at these 2, all right, so this is the newly synthesized from the cycle 1. All right, and they have the 5' end here. All right, and this one also has a 5' end here. And now annealing the cycle 2. Now the other primer anneal to the other side here, and here the other primer anneal to the other side. Then they perform the synthesis so that they will produce a fragment from this end to this end. So that will give you the exact PCR product size, okay? Because now you perform this synthesis so that you will get this strand here. If we go to this picture, so that will be this fragment, all right? That's the end of the cycle 2. You get this newly synthesized fragment and newly synthesized fragment here, okay? So that's the end of cycle 2. You get 4 copies of double helix, and this one and this one are newly synthesized from the cycle 2. Okay, and now when we enter the cycle 3, then again all these 4 double helix will be denatured. After it denatured, then if you look at this one and this one, they'll be here, all right? And this one outside, this one will continue to be the same as cycle 1, and this one will be like a cycle 2, all right? And now this one is cycle 3. Then you can produce the exact the PCR size product, okay? And then after cycle 3, then you have these 8 copies of double helix, so that you can see the amount of the PCR product increase like this. The formula here is 2 to the power of n. N is the number of cycle. So if a 0 cycle, then 2 to the 0 is 1. 2 to the 1, that's when you have 1 cycle, then 2 to the 1, that'll be 2. And then for 2 cycle, 2 to the 2, that'll be 4, all right? And then 2 to the power of 3, for the 3 cycle, and then you get 8 copies. Okay, so this is the amplicon, the PCR product, okay? The amount of the PCR product after the PCR reaction. Okay, and then the primer design. So you have to avoid the inter-strand homologies, avoid the intra-strand homology, and then normally you want to make the forward primer melting temperature as close to the reverse primer melting temperature as possible, okay? So that you can set up the annealing temperature easily. And the product size normally is less than 1 kb, because if you increase the size, and then you also increase the opportunity to bring in the mutant, because when the tech DNA polymerase, when they synthesize the DNA more than 1 kb, it's very likely they can introduce the wrong nucleotide, okay? And as I mentioned, the tech polymerase, which is it's from this bacteria Thermus aquatics, right? So it's the thermostable polymerase, and so it can resist the high temperature. So that's why we use this enzyme to perform the PCR reaction. And then, so because PCR reaction requires a repeated temperature change, and we have to use the thermocycler and then to perform the reaction. And then the interpretation of the PCR result, basically, you can interpret the result by performing the gel electrophoresis to see if the PCR product give you the correct size, right, of the expected size. And that's one way to know whether you get the correct PCR product. And also, of course, it can sometimes misprime, because the primer might adhere to some other region of the template, and then give you the wrong PCR product. So for example, if you have expected PCR product at this size, then you can see the band here. That means they have the misprime, and this is the false PCR product. So this is after geophoresis, you can evaluate your PCR product. And then you can, after geophoresis, you can cut this band and then purify this PCR product, okay, from the gel. For the contamination the control, so basically the physical separation of the pre-PCR and the post-PCR region, so that you have airflow to go to a certain direction, so that you can prevent the contamination. Or you can use a surveillance, you add it to the amplification product plus the UV, so that the surveillance can interact the DNA, intercollect the DNA in the presence of UV, and so they, because they covalently attach to the thymidine, uracil, and the cytidine in the DNA chain, all right, so that they can prevent the denaturation and amplification of the treated DNA, so that the treated DNA will not contaminate your PCR reaction. And then another one, the chemical control, you add the DUTP and the uracil and glycosidase to the PCR reaction, so that they can substitute the TTP, right? Use a UTP to substitute TTP in the PCR reagent mix, so that you can produce the PCR product with the UTP. Okay, and then at the beginning of each PCR, then you add these uracil and glycosilase. You add this to a reaction mix, so that this enzyme can degrade any nucleic acid containing the uracil, so that you can make sure the mixture will be clean, because you add this to the mixture, and the tube they can degrade all the nucleic acid, so that will not contaminate your new PCR reaction. So that's a contamination control. And then to clean the PCR product, okay, so you can do the gel elution. So that means you will perform the gel electrophoresis, all right, and then you cut the gel, and then you perform the purification, all right, from the gel. Or you can perform the solid phase isolation of PCR product. So you just after the PCR reaction, and then you put the whole mixture, the reaction mixture, into the spin column, and then so you can purify the PCR product. Or you can do the DNA precipitation to precipitate the PCR product. Okay, so that's the product cleanup. And then of course there are many applications for the PCR reaction. As I mentioned in the beginning, it's so important because so powerful, and then so many so there are many applications for the PCR. Okay. Any question? That's good. No question. Okay, so I think now I have to go find the previous slide about the PCR reaction, because it seems here doesn't introduce the the calculation of the TM, melting temperature. Okay, so here in this slide, that's in the class 2 slide here, all right? So the PCR reaction here, so basically the primer, okay, the primer. Here is the formula to determine the annealing temperature, all right? So basically, if the annealing temperature too high, no hybridization will occur, okay? So this is because, remember, the melting temperature will be the temperature that you will see 50% of the double strand and the 50% of single strand. When you increase the temperature from the room temperature to 90 degrees, as you increase the temperature, the DNA, double helix DNA will denature, which means they will become single-stranded. And at the point where you have the mixture of the population of 50% double-stranded DNA and 50% single-stranded DNA, then that's called a melting temperature. Okay, so if it's too high, no hybridization, and if it's too low, so that means they can hybridize everywhere. So you can get the non-specific binding. Okay, and then to determine the melting temperature, so this is the formula. Okay, so you can determine the optimum temperature is not 37 degrees. For the 37 degrees, that's the temperature that all the DNA maintain the double helix structure. Okay, for the melting temperature, now be somewhere between 50 and 70 degrees, and at that temperature, then half of our DNA will be denatured. Okay, and then this formula here, okay, you need to know how to use this, because melting temperature Tm equal to 81.5 degree plus 16.6 log m. M is the concentration of the salt in the reaction. Okay, so it could be a normal is a 0.025 mole. All right, so you can do the log 0.025 multiply 16.6 plus 0.41 GC percentage. Let's say the GC is 42%, then you will do the 0.41 multiply 42. Okay, and then you can omit this one, minus 500 divided by the length of the primer. So if the length of primer is 60 nucleotide, then you do the minus 500 divided by 60. All right, so this is the formula that you can use to determine the melting temperature. And then when you set up the annealing temperature, that'll be the Tm minus 2. All right, so this is the very important step. That is correct, Neil. It's a whole number, all right? Not a decimal, like a 0.42. You use the whole number, 42, okay? So this is very important. All right, and the melting temperature, so basically depend on the primer, all right? Because the primer need to bind to the template. So that's why the melting temperature is important. And also during our design, as I mentioned, that you see 2 different TM, all right? 2 different TM in our PCR design. We have the TM1 and TM2. And then you see the TM1 is lower and TM2 is a little bit higher. So that I want you to think about this over the weekend when you design the project that I'm going to assign you as a part of the midterm. So you need to explain me why you see TM1 and TM2, okay? Just follow the principle that the melting temperature depends on the complementary of the primer to the template. Then you should be able to know why there are TM1 and TM2, okay? So you will need to explain that. And then the PCR modification, there are many different modifications, but we are not going to go through every single one, but I will just explain some important one. So like the nested PCR, multiplex PCR, reverse transcriptase PCR, and also the most important one, quantitative real-time PCR. All right, so this is quantitative real-time PCR. Basically, it's the one the whole world is using to determine the presence of COVID-19 virus in the body, okay? So that's quantitative real-time PCR. So for the multiplex PCR, basically that means you have more than one primer, more than one primer pair are added to the PCR reaction, okay? And so that means in normal simple PCR reaction, you only have one pair of the primer. But for the multiplex PCR, then you add more than one primer pair. So it can be two pairs, three pairs. Yes?
I have to leave a little early right now to go to work. Is it okay if I do and I'll look at the recording later?
To do what?
To go to
What
work.
report?
Yeah.
Yeah. Yeah, you can go, but you just need to finish the project, okay? Over the weekend.
The project is due when? The 22nd, right?
Monday morning. It's not the
Oh, it's
22nd. Monday morning.
when the midterms due, right?
Yes.
Okay. So I'll just look at the recording again from this point to the next one. Thank you.
But remember, the class time it's not end so early.
What time does it usually end? 11:30?
Yeah, 11:30, 12 that we finish.
Okay. No, I just have
But
to
it's
come
okay.
and stay and cover someone. That's why. I don't
Okay,
usually.
it's okay. All right, I know this is only the first time you ask. It's okay.
Thank you for understanding. I don't want to take any more time from the class. Thank you, Professor.
Okay. Okay, no problem.
Bye.
So a multiplex PCR, then so you add more than one pair, right? And that's multiplex PCR. So and this is very useful because multiplex PCR normally you can perform several target identification in one shot, so that you don't need to do this several times. Okay, it's very powerful technique. And let me see if there's more example I can show you for the multiplex PCR, because this one is powerful. So like this slide here, the multiplex PCR, so that let's say, because you can do the more than one pair of the primer, then let's say if you have different if mixture of a different sample that you collect, let's say from the environment, you want to know how many different microorganisms in the sample. Then you can have four, let's say in here, you can design 4 different sets of the PCR, one for each specific strain, the different microorganism. Then after you prepare the DNA, then you can add these four pairs of the primer into the reaction, so that when they perform the PCR reaction, if the strain A is in the sample, then you will see a band there. If strain A is not in the sample, then you will not see the strain A fragment. And the strain B, strain C, strain D. So this is an example that you can see. You can evaluate the sample, the mixture of the sample, to see how many different microorganisms in that sample. Or you can purify the genomic DNA from one simple organism, and then from that genomic DNA, you want to evaluate different gene expression, right? So let's say you different for the gene expression, then you can collect the RNA, right? The RNA mixture, and then you can design 3, 4, 5 different genes and primer, and then you perform the reverse transcriptase, and then the PCR, and then you perform the multiplex plus the reverse transcriptase PCR, so that you can perform the reaction in one shot to determine several different gene expression level. Okay, so that's the multiplex PCR. If you combine with a reverse transcriptase, it's very powerful to determine the gene expression. Okay, so that's the multiplex PCR. And the reverse transcriptase PCR. So reverse transcriptase PCR, basically you convert the RNA, all right, into the double-stranded DNA. So the first step would be, I'll show you this PowerPoint again, all right. The first step, again, you collect the RNA, all right, and then the first step for the reverse transcriptase is to synthesize the complementary DNA. It's called cDNA, all right, the cDNA, and then you degrade the RNA from the hybrid, so that you have this cDNA, and then you will synthesize the double-stranded DNA. So this is the reverse transcriptase PCR. Okay, and this is the one that you use to evaluate the amount of RNA. Okay, it's called a reverse transcriptase PCR. And the nested PCR. So the nested PCR, basically, I'll show you the PowerPoint for nested PCR. When you have a low level of a target, all right? When you have a low level of target, and then you want to increase the amount of the target sequence, then you will perform the nested PCR. So nested PCR, basically two-step PCR. All right, two-step PCR. The two-step PCR, you have the first step, and also you have to design two different sets of the primer. Okay. The two different set of primer, because one set of primer will be inside the other set of primer. Okay, so the traditional PCR, so this is traditional PCR like this, but for the nested PCR, so you have two sets, and this is called semi-nested, because the semi-nested, semi-nasty, you have the one primer can be used at one side, and then you have two primer on the other side. For the regular nasty PCR, basically you have one set of primer is outside the target, and then another set will be inside the target. All right. So this example here is a semi-nasty PCR. So basically, for the nasty PCR, the purpose is to basically amplify the DNA first, all right, and then the including the target, all right, and then after the first set of primer finish the amplification, then you add the second set of primer. After you add a second set of primer, they use the product from the first PCR as a template, and then to synthesize the specific target sequence. Okay, so you have nasty and semi-nasty PCR. All right. So that's nasty PCR. And then the real-time PCR, which is the quantitative PCR. And normally it should be written as the qPCR. And the QRT PCR, that means real-time reverse transcriptase PCR. That's called a QRT PCR. Okay. So the real-time PCR basically the difference between the regular PCR and this real-time PCR is the real-time PCR. You need to add a specific dye or the probe that carry the fluorescence, so that you can monitor the amount of the PCR product at the real time. So that's why it's called a real-time PCR. Okay, it's called a real-time PCR. And let me see. Oh, it's here. Oh, I can use this slide. It'd be better to explain. So basically, the PCR reaction, the product will grow exponentially, right? As I show you, 2, 4, 8, 16, 32, and then the signal, all right, can be observed as the exponential curve with a lag phase, log phase, and linear phase, and stationary phase. For the regular PCR, basically, for the standard PCR, you don't evaluate the PCR product until the end of the PCR reaction. Until the PCR reaction, then all you see is the end product of the PCR reaction. Okay, the end product, so that you cannot distinguish the difference between the different sample. You can only see everything reach the maximum. All right. So remember, the PCR reaction is just like the bacteria growth phase. You have lag phase, log phase, stationary phase. So, and then for the PCR reaction, normally you have the lag phase and log phase and then stationary phase. And then for most of the time, the regular PCR, standard PCR, you see the end product, so that you only see at the end the stationary, the saturated product, and you cannot tell which sample has the more starting material. And in order to distinguish the amount of the starting material, like you want to know how much of COVID-19 virus in your body, then you have to use the real-time PCR, because real-time PCR, they just evaluate, they monitor every single cycle of the PCR reaction through the fluorescence, because the real-time PCR, you need to add the dye or specific fluorescence and so that the machine will be able to detect the presence of fluorescence and then give you the curve, right? And to show you how many product has been produced in each PCR cycle, all right? And also there's a threshold, and the threshold basically the threshold is here. Basically, threshold is at the face where it's above the background signal. So because you add the fluorescent signal or dye in the reaction, and the machine always can detect some background, and then when you start the amplify the product, and then the amount of product is proportional to the fluorescence signal, all right? And then when you start seeing the signal above the background, then that's called a threshold, all right? So threshold, and above the threshold, then you will be able to determine the cycle, all right? So that's the real-time PCR. And then, so it's called a CT value, right? The threshold cycle, CT value. And then after that, you can determine the CT value. So for now, for example, they try to define, for example, CT certified, you start seeing the signal. That means you don't have the COVID-19 virus. But if you have a CT10, so that means you have a very high amount of a COVID-19 virus in your body. So the CT value is just like here. So you have the threshold, you start seeing the signal above the background. And then CT certified could be very, very much like still the background value, so that you don't consider the presence of COVID-19 virus. And so in this picture here, so he'll be bigger here, in this diagram here, all right? So you see this red line, okay, that's the threshold cycle. And then for the red line, then you go down to the cycle number. So for example, for this black line, so the CT value is 11, all right? And this green line, the CT value is 14, all right? And then the pink line, the CT value is 31. All right, and how to interpret the CT value. So basically, each one number CT value difference now be 2 times amount difference. All right, so that means, so for example, this black one, let's say CT value is 11, and then the green one CT value is 14. All right, 11 and 14. So that means the initial amount of the DNA of the black curve is 2 to the power of 3 more times more than the green sample. So that means it's 8 times more than the green sample. All right. So because 14 minus 11 is 3, so it's 2 to the power of 3. So it's 8 times different. So this black line has 8 times higher amount of DNA than the green. Therefore, when you see the CT11 compared to the CT14, so CT11 has a high amount of the DNA. And if we use that to determine the COVID-19 virus, so that means this person has 8 times more virus than the other person. Okay? Do you all understand this? Very good. So that's the technique nowadays the whole world is using to determine whether you get the COVID-19 virus or not. All right. And then the detection method, all right? So detection technique for the QRT, the real-time PCR, the qPCR. For the chip detection method is to use the DNA specific dye, and that could be the ethidium bromide, which is very cheap, ethidium bromide, okay? Or you can use the cyber green, all right, cyber green. The cyber green and the ethidium bromide basically will bind the double-stranded DNA non-specifically. Therefore, in the end of each cycle, when you produce the double-stranded DNA, then ethidium bromide will bind. The more DNA, double-stranded DNA you produce, the more ethidium bromide will bind, then you will see an even stronger signal. All right, so, and that's how you monitor the amount of DNA in each cycle. And also you can use a cyber green, right? Cyber green is also a DNA specific dye, but they bind the DNA non-specifically, right? Like this cyber green. So basically, as long as you have double-stranded DNA, the cyber green will bind, all right? So in the end of each PCR cycle, the amount of the signal, all right? The cyber green or the ethidium bromide, they will double. So for example, here, if you have one copy of double-stranded DNA, you have this signal. And then after the PCR reaction, then you double the signal. All right, so that's how you evaluate the amount of DNA at the real time. And then you can also use the hybridization probe. And this will be more specific, DNA specific, and which is more expensive. All right, so you can use a TechMan or the molecular beacon. All right, so these will be a more expensive, but it's more specific, because ethidium bromide, cyber green, they just bind DNA non-specifically. If you have the wrong, if you have the wrong PCR product, they still give you the signal. But the TechMan and molecular beacon, they only give you the signal when you produce the correct target DNA. Okay, so this is TechMan. So you have the standard primer on both sides, and then you have a specific probe, and the specific probe bind to the your target sequence, right? In between these two primer, and this probe carry the reporter dye and quencher dye. So when they bind to the during a annealing stage, when they bind to the template, there's no signal. But it's because the reporter dye is quenched by the quencher. But after the synthesis, all right, after the synthesis, then this probe will be degraded, all right, because the enzyme, the polymerase, has a 5' to 3' exonuclease, so they can degrade this probe. As soon as they degrade this probe, then the reporter dye show the signal, all right, during the DNA synthesis, the step 3, then you will observe the signal. And then the more PCR product you produce, then the more signal you will observe. All right, so that's the reporter dye TechMan, and molecular beacon is a similar idea. All right, it's just a different way to quench the reporter dye. So that's basically the important modified PCR reaction. All right. So that's the so that's basically for today's material. Okay.